and it is called a dark band, and this is called light band. Type structure started, skeletal muscle and cardiac muscle. Clear? And in case of skeletal muscle, they comprise of long muscle cells. Due to their length, they are called fiber, tontu, fiber. তন্তু বায় বলে না, তন্তু বায়, হ্যাঁ, তাঁতি, সেই জন্য ওই যে ফাইবার নিয়ে কাজ করে, সুতো নিয়ে কাজ করে। দে আর কল মাসেল ফাইবার্স, elongated structures, long structures. It may be 30 centimeter or so in case of long muscles, or maybe few millimeter in case of small muscle. Multinucleated. In case of skeletal muscle, cardiac muscle, this type of structure, and they are formed of this type of muscle cells or muscle fiber, muscle fibers. Muscle fiber. They are also this type of structure seen in this case, muscle fiber. And the muscle fiber diameter is 20 to 100 nanometer, muscle fiber. And a muscle, skeletal muscle is an organ. And when you have seen the muscles in your anatomy department, they are formed of fascicles. Fascicles are formed of individual muscle cells, and individual muscle cells are comprised of myofibrils. And the fibrils contain the filaments, and filaments are formed of molecules. In this way they are organized. বোঝা গেল? Muscle, in all the textbooks, they have this type of figure. Biochemistry text, physiology text, muscle, total muscle, and it is an organ. It comprises of multiple tissues. It contains nerves, it contains blood vessels, and the muscle cells itself. They are the structural unit, muscle cells or muscle fibers. But how they are, if you take the muscle, any muscle, biceps muscle, deltoid muscle, water muscle, then it is formed of muscle fascicles. Fascicles are formed of muscle fibers, and muscle fiber is composed of myofibrils. Myofibrils are formed of filaments, and filaments are formed of the molecules, actin and myosin. Suppose you have a cross section in this way. If you take a cross section of this muscle cells, and it may be 30 centimeter, or in case of millimeter, it is variable. And they are not connected to each other by any gap junctions, so they can function individually. And you have to take the cross section, then you will find that these type of structures, they are also elongated structures. 1 to 2 mu in dimension, and these are myofibrils. And these myofibrils, they contain these filaments. And this arrangement of the filaments is responsible for this type of these striations. In the light microscope, up to this resolution is possible, but with electron microscope and other sophisticated methods, further details has been known. X-ray crystallography and this. Now this is the myofibril, it is the muscle cell or muscle fiber, and it is this, they are, and these structures, they are the myofibrils. Myofibrils are formed of these filaments. Now these filaments are arranged in such a way, and they are registered in such a way that this type of structure is possible. If they are not arrayed in regular way, if there is overlapping, then it will obscure this type of striations, but it is not. Suppose in case of myofibril, you take a myofibril in this way, you will find, then you find that they are disposed in this way. Thick filament, and intermediate, this is the thin filament. Thin filament. These are thin filaments. On this side also, there is the thick filament. They are arranged in this way. And muscle length is variable muscle length. It may elongated, it may contract up to 2/3 its original length. Suppose in case of a muscle, if you consider it is the resting condition, and when it is contracted, its volume will increase, but the length will diminish. Volume will increase. It is the resting condition and it is the contracted condition. Clear? In this condition. So, when there is any blood vessel that is interposed between two neighboring skeletal muscle, the contraction of the skeletal muscle can close its lumen, approach its lumen. Muscle pumping effect of blood vessels. Effect on gravity or so, contraction of the muscle, venous return, muscle pumping effect. How it is possible? Clear? Suppose there is a blood vessel. It is the blood vessel in the relaxed condition. Now, if there is contraction, then it will approach in this way, and it is approaching this way. And then the space in between the walls will diminish. There is narrowing or even obliteration in between them. Obliteration. And when it is on the venous side, so whenever it is completely open, then there is flow. But when it is obstructed, there is no flow. And when there is expansion, then relaxation, then the flow will increase. In this way, they will have an effect, muscle pumping effect. And there are venous valves opening toward the heart. In this way, with exercise, the blood will come from the lower end. যদি স্ট্যান্ড স্টিল হয়ে কেউ দাঁড়িয়ে থাকে, তাহলে পরে কী হয়, অনেক সময় দেখা যাচ্ছে যে ব্লাড ভলিউম ডিউ টু গ্র্যাভিটি accumulates in the lower part of the body, and the venous return may be low, and there is fall in the cardiac output, stroke volume, and even an comatose stage or loss of consciousness may be there. Postural hypotension. বুঝতে পারছো? এরকম হতে পারে। It is the muscle pumping effect. Now we consider the structure, how they are produced. It is the myofibril and it is the filaments, thick filament and thin filament. Structure of these filaments, and these are cytoskeleton, part of the cytoskeleton present in other cells. You are familiar with the cytoskeleton, and these are molecular motors, cytoskeleton, microtubule, microfilament, intermediate filament, you are familiar with. Kinesin, dynamin, dynein, you are familiar with. So these are in reality they are these contractile proteins or the structural proteins they are. And it is the actin filament or microfilament. This actin filament is similar to that of the actin filament. Actin and it is the myosin. Myosin filament is forms of myosin molecule. Myosin filaments are formed of myosin molecules. Myosin molecules, they are aggregation of this. These myosin molecules are heterohexamers. I am coming one by one. Heterohexamer. It consists of, that is the 4 light chains and 2 identical heavy chains. They are in this way. Suppose with another color, twisted in this way. And contains 2 light chains. C terminal and it is the C terminal. And these are these heavy chains and they are twisted in this way. It is the N terminal and it is the C terminal. And they together form the head part. And these are the light chains. 4 light chains. Of these 2 are essential light chain and a regulatory light chain. Or in some case there is a phosphorylatable light chain and alkali light chain. And this head part really binds with the binding site with actin. It has also phosphorylation site. And they form cross bridges. And due to movement of these cross bridges is responsible for the contraction of the muscle. Clear? And hundreds of these unite. There are 2 heads and one head operate in a single time. And hundreds of them accumulate or they aggregate to form this. And how this aggregation is possible? If you take a short stretch, suppose how they are disposed. These are all easy things and in this way. Hundreds, they unite you see the change in polarity. They aggregate in this way and they together form this thick filament. The dimension is 16 nanometer or so. In some case 12 nanometer, whatever it may be. It is more than that of the actin filament. In this way they are disposed. And at the very central part, then it is free from these side chains. Central part. In this part, it is free from the side chains. And they form the M line. And there is this reversal of the polarity. বিপরীত দিকে থাকছে, reversal of the polarity. And intermediate part, they have some swellings and they are connected by means of some filaments, structural filaments. And the center part, they do not bear the cross bridges, and there is reversal of the polarity, and they are called M line they do form. Clear? In this way, they are formed. So, myosin molecules, they are heterohexamers. It contains two heavy chains, and it is the tail part and it is the head part of these heavy chains, and four light chains. Four light chains. On either side, one is the phosphorylatable light chain, or alkyl light chains, or essential light chain, and a regulatory light chain. And this is the head part that from the cross bridges, and it is the functionally important. It can unite with the actin, and it has been, and its length in reliable test, say they up to this 134 nanometer, and this part is 9 nanometer, head part 9 nanometer, and they can be split by different types of enzyme, and by means of, suppose it is trypsin, by means of trypsin, TRYP, trypsin. You are familiar with the trypsin? ট্রিপসিন কোথায় পাওয়া যায় বলো, ট্রিপসিন কোথায় পাওয়া যায়? তুমি তো পড়েছো, পরীক্ষা দেবে তোমার। ট্রিপসিন বসে বলো, ওসব করতে হবে না। ট্রিপসিন কোথায় পাওয়া যায়? এই তুমি বলো, ট্রিপসিন কোথায় পাওয়া যায়? বসে বলো, ওই তো কষ্ট করতে হবে না। হ্যাঁ, প্যাঙ্ক্রিয়াসে ট্রিপসিনের প্রিকারসার পাওয়া যায়। বলতে হবে যে ট্রিপসিনের প্রিকারসার, সেটা ট্রিপসিনোজেন, সেটা সিক্রেটেড হয়, কিন্তু ট্রিপসিন উৎপত্তি হয় স্মল ইন্টেস্টিনে। স্মল ইন্টেস্টিনে সেখানে ট্রিপসিন পাওয়া যায়। কাজেই বুঝতে পারছো যে এই যে পরীক্ষা, এই যে একে প্রশ্ন করলো ও করতে পারছে না, ও এখন বুঝতে পারছে, এখন বুঝতে পারছো জিনিসটা। তোমাকে যদি কখন পোড়ালে প্রশ্ন করে, তখন তুমি চিন্তা করবে যে এরা লোকটার প্রশ্নটা করলো কেন, করলো কেন। খুব সহজ সাবজেক্ট। তখন বলবে যে ট্রিপসিনোজেনের উৎপত্তি হচ্ছে প্যাঙ্ক্রিয়াসে। তখন আমি জিজ্ঞাসা করবো প্যাঙ্ক্রিয়াসের কী টাইপের সেল থেকে সেটা বেরো, তখন কি বলবে? অ্যাসিনার সেল। মানে বুঝতে পারছো এখানে নাম্বারের ভ্যারিয়েশনটা হচ্ছে, তার মানে ওর কনসেপ্টটা পরিষ্কার আছে। কিছুই কনসেপ্ট নেই, সহজ সাবজেক্ট। এই জন্য যাই হোক, তা এখন এই জন্য এই অ্যাসিনার সেল থেকে, তার মানে সিক্রিশনের হচ্ছে, তার মানে এটা কী ধরনের সিক্রিশন? এটা হচ্ছে এক্সোক্রিন সিক্রিশন। তার মানে এর পাশের উপযুক্ত হয়ে গেছে, বুঝতে পেরেছো? এরকমভাবে যদি পড়ে যায়, ভালো রেজাল্টই ও করতে পারবে। এই ভালোই পাশই, এখন ও ইন্টারন্যাশনালে পাস করে গেছে আমার কাছে, বুঝতে পেরেছো? আর ইনসুলিন কোন সেল থেকে তৈরি হয়? বিটা সেলস। কোথায় থাকে এটা? ইন্টারস্টিশিয়ামে থাকে। তার মানে কি ভালোভাবে পাস করছে। ওকে 80% দিতে আমার কোনো রকম দ্বিধা হবে না, কি 10%ও দিয়ে দিতে পারি। সেই রকম, বুঝতে পারছো? সহজ সাবজেক্ট এগুলো। এই জন্য তুমি ঠিক বলেছি না, ক্লাসের দরকার নেই বলে তোমার মনে হয়তো না, আছে, দরকার আছে এখনো। So this myosin is produced. These are the myosin molecules. Clear? Myosin molecule. And it is this myosin molecule, its molecular weight is more than that of the actin, myosin molecule. কাজে এর হচ্ছে মনে করো 260,000 মলিকুলার ওয়েট, 260,000 ম্যাসিন মলিকুল যেটা থাকছে, এই একটা মলিকুল, এটা 260,000, এটা দেখে নেবে বই থেকে, বুঝতে পেরেছ? নাও দিস অ্যাক্টিন, হাউ দিস অ্যাক্টিন ইজ প্রডিউসড? অ্যাক্টিন ইজ প্রডিউসড ফ্রম গ্লোবুলার অ্যাক্টিন। দুটো চেন নিয়ে যদি তুমি পুঁতি থাকে না, পুঁতির কাজ করেছে যারা, তোমাদের মধ্যে কেউ পুঁতির কাজ জানো না, পুঁতির কাজ করতে পারো না? ওই যে সুতোর মধ্যে ছোটো ছোটো গ্লোবিউল সমস্ত দেয়, এরকম দুটো লাগালে, দুটো তৈরি করলে তারপর যদি জড়িয়ে দেওয়া যায়, তাহলে পরে যেটা তৈরি হয় সেটাই হচ্ছে অ্যাক্টিন ফিলামেন্ট। জি প্রোটিন, ইট ইজ দি সলুবল প্রোটিন, বাট হোয়েন ইট পলিমারাইজ, দেন ইট বিকামস ইনসoluble in presence of magnesium, and with or without help of enzyme or so, and it is suppose in this way. In this way, they polymerize. This around they are called G proteins, and also in this way. In this G proteins, and molecular mass is 43,000, 43K, or is the molecular weight. They form the G, and the total form is the F actin, actin filament, actin filament. Actin filament. And in this case, if you see, there is the group between the group. পুঁতি যদি তুমি জড়িয়ে দাও, যদি আঙুলটা জড়িয়ে দাও, তাহলে দেখবে গ্রুপ তৈরি হচ্ছে। নাও উইদিন দিস গ্রুপ দেয়ার আর ট্রোপোমায়োসিন। বুঝতে পেরেছো? যেমন একটা আঙুল দিয়ে যদি একটা আঙুলের সঙ্গে জড়িয়ে দাও, তাহলে গ্রুপ তৈরি হবে, খাঁজ তৈরি হবে, তাহলে দুপাশে দুটো খাঁজ তৈরি হবে, নিজে বুঝতে পারবে। সুতো নিয়ে বড় মোটা একটু দড়ি মতন নিয়ে যদি পাকিয়ে দেখবে নিজে, ফিজিক্যালি সেটা দেখবে দুটো গ্রুপ তৈরি হচ্ছে এপাশ ওপাশ করে। তাহলে এই গ্রুপের মধ্যে কী থাকছে? ট্রোপোমায়োসিন। And within this group, there is the tropomyosin, elongated molecule, in this way. Double, alpha, beta, in this way. Tropomyosin, they connected to each other. Suppose it is alpha, it is beta, it is alpha. In this way, they are connected. Tropomyosin molecules. They form the tropomyosin protein at the groups on either side, tropomyosin. And their molecular weight is 70,000, tropomyosin. Two chains in this way. They unite in this way. Elongated molecules. Clear? Suppose it is alpha, it is beta, it is alpha. It can communicate with it. In this way, they are produced. Tropomyosin. And at distances, suppose the distance is 38.5 nanometer or so, and there are these globular molecules. They are called troponins. Have three components: troponin T, troponin I, and troponin C. Suppose at these distances, there are this type of molecule. It is the troponin T that binds the other part with it. And there are troponin T, troponin I, and troponin C. Suppose it is the troponin T, troponin I, and it is, suppose it is the troponin C, another troponin C. In this way, they are, and their molecular weight 17,000 to 35,000. They are molecular weights. Troponin I, troponin T, troponin C. Troponin T binds with the tropomyosin, and troponin I is placed in such a way that it obscures the site at which the myosin molecule should attach. Suppose it is obscuring, in the resting condition, it is obscuring this site. It is obscured by the troponin T. And this side, the myosin head can bind in the resting condition. Inhibitory, I means inhibitory. And when the troponin C binds with calcium ion, books differ the number of the calcium ion. Common physiology text, they only mention single calcium ion. When it binds with it, and biochemistry text like 4 calcium ion, whatever it may be, everything is true. When it binds with the calcium ion, then there are some conformational change, and there is this attachment of the troponin I with the actin. Troponin I with the actin is loosened. It becomes weaker, and at the same time there is some lateral movement of the tropomyosin molecule, and it is obscured by this troponin T. So it is obscured by the troponin C, troponin I, troponin I. But when calcium ion binds with it, when calcium ion binds with troponin C, then there is some weakening of this troponin bond between the troponin I and actin, and there is a movement or lateral movement of the tropomyosin molecule, so that this binding site that has been obscured and it has been exposed. Suppose there is the movement, it is from this position to this position, and this space with which the actin should bind, and it will be exposed. It will be exposed, and myosin head can bind with it. Myosin head can bind with it. Clear? And cross bridge, normally there are cross bridges, but they are in the cocked position. They are not attached, and in the resting condition there is no net movement. Suppose in rough pictures you can understood, suppose it is the actin filament, and it is the myosin, particular part of the myosin molecule, and it is attached in this way. It is just short of not attaching in this way, and there is the site where it can bind. It can bind. It is the actin filament, and it is a single head of this. There is another head I am not mentioning, and it is the myosin. Suppose it is the myosin filament, and from its projection there is the head part is projecting, and there is a gap between this actin and myosin normally in the resting condition. And whenever there is the arrangement is that the calcium ion do enter into the cytoplasm, and then this reaction is possible. Clear? And for this movement, this liberation of energy is required. It is a chemical transducer. ATP is broken down into ADP and PI, and this head of the myosin, it has ATP binding site. Textbook does not mention exactly whether it is light chain or whether it is part of the heavy chain, but from the discussion it appears that this part of the heavy chain, there are some area that has a myosin ATPase activity. Clear? Activity. And this activity is increases many times when actin binds with it. বুঝতে পারছো? বায়ুবর্ণী যোগের মতন আগুন আছে, কিন্তু যদি বায়ু যদি আসে, ক্যালিফোর্নিয়াতে যেমন হচ্ছে, বায়ুটাকে আটকাবার চেষ্টা করছে, বলে না বায়ুবর্ণী সংযোগের মতন, সেটা তখন তীব্র গতিতে বেড়ে যায়। বুঝতে পারছো তো জিনিসটা? বায়ুবর্ণী, সেজন্য সেরকমভাবে যেই অ্যাক্টিন এসে যুক্ত হলো তখন সেই আগুনের মাত্রা বেড়ে যাবে, এনার্জিটা বেড়ে যাবে। দেন দিস ADP and PI will be liberated, and there is the power stroke. There is the movement of the head with the neck. You can, from this simple picture, you can find it is the tail part. Suppose if you further it, there is the head, and so it is the neck part, it is the head part, it is the neck part, and it is the tail part, you can consider. Then there is the movement is possible. And for this purpose, there is a requirement of calcium ion. Within the resting muscle, skeletal muscle, calcium ion concentration is very low. It is value. What is the value concentration of? What is the concentration of calcium within the skeletal muscle, resting skeletal muscle? -7? হ্যাঁ, এই তো বুঝতে পারছো? তার মানে আমি আগের দিন আলোচনা করেছিলাম, তুমি শুনেছিলেন না সেটা? তুমি আগের দিন ছিলে? তাহলে 10⁻⁻7 মোল পার লিটার, 10⁻⁻7 মোল পার লিটার is equal to 100 into 10⁻⁻⁹ mole per liter or mole, and it is the nanomole, so it is 100 nanomole. But it is easy to remember this mole per liter. And when it increased to a value of 10 to the power minus 5 mole or mole per liter, then this contraction is possible. And this calcium can enter within the cell by which mechanism? By means of calcium can enter. Calcium can enter. There may be ligand gated channel, there may be voltage gated channel, there may be store operated channel, different types of, in this way calcium can enter. And calcium can be removed by other pumps and also exchange with sodium. Here. Now how within this skeletal muscle calcium must do increase, and it is a quick process. যেমন আমি ভাবছি এটা হাতটাকে করব, ফ্লেক্স করব, সঙ্গে সঙ্গে আমি করতে পারছি, মানে ফ্র্যাকশন অফ এ সেকেন্ড এটা করতে পারছি আমি। তাহলে সেই সময় ক্যালসিয়ামটা অ্যাভেলেবল ছিল, এই অবস্থায় অ্যাভেলেবল নেই, কিন্তু যেই আমি ভাবছি এটা তুলব সঙ্গে সঙ্গে করতে পারছি। সো ইট ইজ এ ভেরি কুইক প্রসেস, অ্যান্ড হাউ দিস উইদিন দি শর্ট টাইম দিস ক্যালসিয়াম বিকামস অ্যাভেলেবল। So there are the structures that are called sarcotubular system. Sarcotubular system consists of T tubules and sarcoplasmic reticulum. Clear? Sarcoplasmic reticulum, sarcotubular system. And what are they? But before going into that, we have to mention something part remained, that is this line that transects this myofibrillin in between the two actin filaments, and it is called Z line, or Z disc you may call Z line. It is the Z line on other side, Z line. And it is and this type of bands are seen, and this together on either side, and it is called A band or anisotropic band, A band or anisotropic band, simply dark band, and this band on this side and this side, and so it is this I band. I by 2 in this side, I by 2. In the relaxed muscle, I band, isotropic band, isotropic band. And it is due to differences in refractive indices. What is your concept regarding isotropic and anisotropic? তোমাদের কি মনে হয়? Isotropic and anisotropic? হ্যাঁ হ্যাঁ কি বলো, বলো না, পরিষ্কার করে বলবে, আমার জ্ঞানও খুব সীমিত। হ্যাঁ কি পরিষ্কার করে বলো, আর একটু পরিষ্কার হোক, সবাই শুনুক, তো শুনুক, তুমি বলো পরিষ্কার করে। আইসোট্রপিক বলতে কি বোঝায়? হ্যাঁ শুধু অ্যাক্টিন না, বসো, ঠিকই বলেছে, এটা হিস্টোলজির ক্ষেত্রে আই ব্যান্ড শুধু যেখানে অ্যাক্টিন ফিলামেন্ট আছে সেই জায়গাটা আইসোট্রপিক। আইসোট্রপিক মানে হচ্ছে কোনো একটা সাবস্টেন্স, মনে করো গ্লাস, তার যে কোনো ডিরেকশনই তুমি যদি মেজার করো দেখবে রিফ্র্যাক্টিভ ইন্ডেক্স সমান, মানে তার সব সমান আছে। কিন্তু অ্যানাইসোট্রপিক হচ্ছে কি, তার ডিফারেন্ট প্লেন অনুযায়ী, ডিফারেন্ট ডিরেকশন অনুযায়ী তার রিফ্র্যাক্টিভ ইন্ডেক্স ভ্যারি করে, সেটা হচ্ছে অ্যানাইসোট্রপিক। ও যেটা বলেছে হিস্টোলজি অনুযায়ী ঠিকই বলেছে, যেখানে আই ব্যান্ড এবং এ ব্যান্ড আছে এবং অথবা শুধু এ ব্যান্ড আছে, সেখানে ডিফারেন্ট ডিরেকশন থেকে আলো গেলে তাদের রিফ্র্যাক্টিভ প্রপার্টি ভ্যারি করে। এটা অন্য কিছু প্রপার্টির ক্ষেত্রেও এটা ব্যবহার হয়, আইসোট্রপিক অ্যানাইসোট্রপিক, অন্যান্য প্রপার্টির ক্ষেত্রে, মানে একটা হোমোজেনাস আর একটা নট হোমোজেনাস সেরকম। এইটা ভালো করে দেখে নেবে, এটা তোমাদের ডিউটি হচ্ছে এই থেকে দেখে নেবে, ইউটিউবের তোমার গুগল থেকে দেখে নেবে আইসোট্রপিক অ্যানাইসোট্রপিক, পড়লে ভালো বুঝতে পারবে, এবং অনেক এক্সাম্পলও দেওয়া আছে সেরকম। কমন সেন্স দিয়ে বলা যায়, তুমি যদি গ্লাস নাও তাহলে পরে দেখবে গ্লাসের যে কোনো যা ডিরেকশনে তুমি যদি নাও তাহলে দেখবে তোমার সেখানে রিফ্র্যাক্টিভ ইন্ডেক্স একই পাচ্ছ। কিন্তু কোন ক্রিস্টাল বা এরকম আছে ক্যালকাইট ক্রিস্টাল বা ক্রিস্টাল ডিফারেন্ট ডিরেকশনে লাইটের হচ্ছে রিফ্র্যাক্টিভ ইন্ডেক্স ডিফার করছে, সেটা তার স্ট্রাকচারের উপর নির্ভর করছে এবং সেটা বলি অ্যানাইসোট্রপিক। এখানে আমরা অতটা মনে রাখি না, আমরা রিফ্র্যাক্টিভ ইন্ডেক্সের চেঞ্জের জন্য আমরা বলি এই এ ব্যান্ড হচ্ছে ডার্ক এ ব্যান্ড হচ্ছে অ্যানাইসোট্রপিক, আর লাইটের সেকেন্ড লেটারটা দিয়ে মনে থাকবে তোমার আইসোট্রপিক, সেকেন্ড লেটারটা দিয়ে। এটা হচ্ছে সেকেন্ড লেটার অ্যান্ড ইট ইজ দ্য সেকেন্ড লেটার আইসোট্রপিক অ্যান্ড ইট ইজ দ্য অ্যানাইসোট্রপিক। Now this gap between the Z lines, this line, and it is called sarcomere. It is a functional unit, sarcomere. And you see within this dark band there are some, to some extent, some lighter band where there is no interdigitation, only thick filament, and it is relatively lighter. Even within the dark band there is the middle part, there is the lighter band in the relaxed state, and they form the edge band. And within the edge band there is some swelling and it produces the M line. And in the previous figure you have noticed that just outside the M line there is no cross bridges, no head, and it is free of that cross bridges, and it is called, so to some extent, lighter, and it is called pseudo edge zone. Very close to this there is the, where there is no, they together they form the pseudo edge zone. Because there is the absence of the cross bridges at that very narrow pavement. Just close to the midline on either side there is a relatively lighter area within this edge zone, and it is called pseudo edge zone. So in the relaxed muscle edge zone is present, but whenever there is contraction then they will overlap. And what is the dimension of this sarcomere? In the contracted state it is 1500 nanometer, and whenever it is completely relaxed it is separated 2300 nanometer. So it is equivalent to 1.5 μm, it is 2.3 μm. এগুলো সব দেখবে নিজেরা বুঝতে ক্যালকুলেট করে ভুল হয়ে পড়ে সেটা আমাকে বলবে আপনার এমনিতে জায়গাটা ভুল হয়েছিল, বুঝতে পেরেছেন? তাহলে পরে তোমাদের নলেজটা থাকবে। ইন দিস ওয়ে দে আর ডিসপোজড clear. And whenever this troponin, this T system is present at distances, another type of system, and whenever there is calcium ion is acting, then it is the sliding movement of this tropomyosin and 7 binding sites are exposed. When calcium works due to the lateral movement of the tropomyosin and there is 7 binding sites are exposed, and it is organized in such a way that minimum intervention is required, and they are placed at distances. Suppose another T system, another troponin system, it is in the 38.5 nanometer. And in case of actin filament, that is this periodicity is 35.5 nanometer. Paracrystalline fashion they are disposed. Whenever there is calcium attachment with the troponin C on one side, and there is movement and there is exposure of the 7 binding sites. The nature has made this so that so much quickly it is possible. Yeah. And for this calcium must be available quickly, and it has to be reabsorbed quickly so that the quick movement is possible. So there is the term, there is the excitation contraction coupling, and what it is and how it is possible. sarcotubular system is related to this sarcotubular system. That is at the AI junction, that is the anisotropic isotropic junction, thick filament. On either side. It is the thick filament and it is the thin filament. Even in the relaxed muscle, whatever. And they are all in register. That is, whenever there is muscle cells are present, there are innumerable number of these myofibrils. In spite of this, every myofibril and every cells, they are in a register. They are opposite to, there is no overlapping in that sense. Otherwise, how this striations is possible? And that developmentally, they are geared in this way, Now, at the AI junction, it is the A, it is the I junction. It is the AI junction. It is the AI junction. At this point, there is this prolongation of subtubule from the sarcolemma tubule. And tubular vesicle-like structures by electron microscope. There is prolongation it from the sarcolemma. At the periphery of the muscle cells, there are numerous mitochondria. And they are functionally, they are every individual cell you may consider the syncytium. Multiple nuclei, and there must be the adequate number of T tubules so that communication can reach them quickly from the cell membrane. And the cell membrane of skeletal muscle is called sarcolemma. And it is the transverse tubule or T tubules. On either side of it, and then there is another T tubule at the AI junction. It is coming from sarcolemma. It is continuation of the extracellular space. The sarcolemma is surrounded by some basement membrane, and this continuation of the sarcolemma is as a T tubules. It is a view of one section, profile view, and you have to consider as a disc. You have to consider as a disc in this way. There are perforations, and this perforation through which the myofibril traverses. Then the concept will be clear. Perforations. They are crossed by these myofibrils. They are crossed by the myofibrils. This is called disc-like structures, perforated by the myofibrils. And within this, and besides this, then there are some structure, curtain-like structures, curtain, C-U-R-T-A-I, incomplete curtain-like structures. These are all tubules and vesicle-like structure. They from the sarcoplasmic reticulum, derived from the endoplasmic homogeneous, it is similar to that of the endoplasmic reticulum, smooth surfaced endoplasmic reticulum. And in this case, then at this part, there is some dilatation on either side. On either side. So you see, these are these cisterns or dilatations where there is accumulation. It is so. It is at this time there is the accumulation of calcium at the site. Terminal cisterns, they are called. And they together form the triad. Triad. T tubules means this is the transverse tubule. It is the invagination of the sarcolemma. So this lumen is connected to that of the extracellular fluid. It is connected to extracellular fluid. And the sarcoplasmic reticulum, it has the terminal system that opposes the T tubules on either side, and they form the triad. And the rest of the part, it is the longitudinal tubule, L tubule. Longitudinal tubule, L tubule. Now action potential is coming in which way? Action potential is produced in skeletal muscle at which site? Myoneural junction, muscle end plate. It is included in your higher secondary syllabus. আমার মনে হয় হায়ার সেকেন্ডারি সিলেবাসে এগুলো তোমার দেওয়া আছে, তোমরা নিশ্চয়ই পড়েছো। হ্যাঁ, মাসেল এন্ড প্লেট, হ্যাঁ পড়েছো না? হ্যাঁ, মাসেল এন্ড প্লেট, muscle end plate. Suppose a nerve is coming and it is this neuromuscular junction or motor end, and it contains this type of structure to increase the surface area, and there are neurotransmitter present within this neurotransmitter. Which neurotransmitter is present in these vesicles? Which neurotransmitter? Acetylcholine থাকে, হ্যাঁ, only acetylcholine. And acetylcholine is released in response to action potential coming from the anterior horn cell. Voluntary muscle. Skeletal muscle is voluntary muscle. We are speaking everything, and our life is responsible for the skeletal muscle. Skeletal muscle. Udgid bola hai. Skeletal muscle. Life is responsible. Because respiratory muscles are skeletal muscles. We are normally not conscious of our respiratory processes, but it is skeletal muscle, and you can have change in the process of respiration. You may stop breathing for some time. You may enhance the breathing, and it is required for different types of sound production. Sound এর সঙ্গে তো যোগ আছে। Expired air, by the expired air there is a vibration, and different vibration is responsible for a melody. It may be rough like my voice, that is disliked by everyone I came across my life, and today of course, but it is melodious everywhere. Now acetylcholine is liberated, passing through the synaptic cleft, it combines with muscle type acetylcholine receptors, and it is called, which type of receptor it is? It is the nicotinic receptor. By which subunits they are formed? It is the one example of, that is the transport across the cell membrane. Ligand gated ion channels, we discussed in one class. Ligand gated ion channel, a typical example, nicotinic cholinergic receptor formed of 5 subunits. Now whatever it may be, when this calcium ion when acetylcholine do enter in this, nicotinic receptors are there, and they become stimulated, and then there is this entry of the sodium ion and potassium ion, and by the sodium ion initially there is formation of motor end plate potential, and regularly it crosses the threshold level. Then action potential will be generated, and it will spread through this other part of the sarcolemma, and by means of T tubules, and they will enter. And there are activation of the two types of receptor here. In this case, voltage gated DHPR, dihydropyridine pyridine receptors, voltage gated calcium channel, slow calcium channel receptors. Some calcium channel can enter and may have function, but within the skeletal muscle it is not required. And there is the ryanodine receptor, RYR receptors, ryanodine receptors, and it is the ligand gated calcium channel. When calcium unites with it, or in this case skeletal muscle, external concentration of calcium is not essential, and there is some physical process by means of which calcium will be liberated and it will have its action. Time is up, so we should stop, and next we start from this part onward. Purono pora ikore neওয়া যায়. Contractile proteins are actin and myosin, similar to other structural proteins. Actin and myosin. Myosin is formed of myosin molecules. One alpha and another beta chains. They are placed in this way. And at distances of 38.5 nanometer, there are troponin complex. It is the globular unit, and they are troponin I, troponin T, troponin C. Troponin T binds with the tropomyosin, and troponin I binds with the actin. And normally it obscures the site where there is the myosin head can unite. Clear? And when calcium ion binds with the troponin C moiety, then there is weakening of this bond of troponin I and actin. And there is slight movement of these tropomyosin filaments by conformational change. And then the head unites, and then there is this power stroke, liberation of ADP and PI, and there is movement of the head part when this is 45 degree, this type of movement. 10 nanometer the distance in this case, and in this way, this actin filament will move within these myosin filaments. So individual filaments have their length fixed, but this H band will disappear, and the I band will almost completely disappear in complete contraction. Clear? I think it is all for today.